Good morning everyone. Today topic photorespiration, also called P2 cycle. Also it has another name glycolate pathway, or also it has another name oxidative photosynthetic carbon cycle. So this photorespiration concept was proposed by a scientist called Warburg. Proposed by Warburg. So generally photorespiration is a waste process occurring light, means photo. And in this photorespiration process involves the consumption of oxygen with release of carbon dioxide. Here note, generally in photosynthesis, what happens? Consumption of carbon dioxide with release of oxygen, but it is quite opposite. So here I am writing note, important note here. Usually in photo, consumption of carbon dioxide with release of oxygen will occur, but in photorespiration, consumption of oxygen and release of carbon dioxide will occur. So here why this process is wasteful process means because of, unlike photosynthesis, it loses, very important, it loses fixed carbon. It lost the fixed carbon in the form of carbon dioxide. It loses the fixed carbon in the form of carbon dioxide. It lost the carbon dioxide instead of fixing it. Actually, generally in photosynthesis, carbon dioxide must and should be fixed. But here, the carbon in the form of carbon dioxide, the carbon is lost in this photo instead of fixing it. And without producing any kind of chemical energy like ATP and NAD. So without producing any chemical form of energy like ATP and NADP, this photorespiration process, it loses fixed carbon in the form of carbon dioxide instead of fixing it. So why does this photorespiration process occur generally means we will discuss. Why does it happen? Why does photo? So the main causes or culprit or reason for this photorespiration occur in C3 plants, that is due to an enzyme called RuBisCO. Due to RuBisCO enzyme, very important enzyme. So due to RuBisCO enzyme, this photorespiration process will occur in C3 plants. Why means generally about RuBisCO enzyme. So RuBisCO is the most abundant enzyme in the world. Very important point here. Very important. So RuBisCO is the most abundant enzyme in the world. So here RuBisCO full form ribulose 1,5 bisphosphate. Here C means carboxylase and oxygenase. So this enzyme has dual functions. It has dual function. Very important. So it can bind, if it can bind the oxygen, then it's said to be as a oxygenase, or it bind to the carbon dioxide, then it's said to be as a carboxylase. Means this RuBisCO has dual function like oxygenase and carboxylase. So here generally this RuBisCO has active site for both. So this RuBisCO has active sites. It has active site to bind with oxygen and carbon dioxide. So generally this RuBisCO has much higher affinity. Usually RuBisCO has much higher affinity for carbon dioxide, very important, carbon dioxide, then oxygen usually. So RuBisCO has much higher affinity to bind to carbon dioxide than oxygen. Here one important term, the relative concentration of oxygen and concentration. The relative, here the concentration of oxygen and carbon dioxide, which determine which one will bind to the enzyme active site. So the concentration of oxygen and carbon dioxide only which determine the which one, means either oxygen or carbon dioxide should bind to the active site of enzyme. For example, here I will write another slide. Yeah. For example, if carbon dioxide concentration is more, then RuBisCO act as carboxylase enzyme and which bind to the carbon dioxide. How means like this. Here generally this RuBisCO bind the carbon dioxide to the RuBP. Here RuBP will write. Yeah. So here RuBP means ribulose 1,5 bisphosphate. It is a 5 carbon compound which bind to the carbon dioxide. When this RuBisCO act as a carboxylase means if the carbon dioxide concentration is more. So in presence of this RuBisCO enzyme, which act as a carboxylase enzyme which bind to this, and then it involved in formation of 2 molecule of 3-PGA phosphoglyceric acid. So it is this 3-PGA involved in C3 cycle or Kelvin cycle. C3 or Kelvin cycle. If oxygen concentration is more, means if oxygen concentration is more, then this RuBisCO enzyme which act as oxygenase enzyme, then it binds to the oxygen. For example, how means like this. Yeah. So here RuBP 5 carbon compound which bind to the oxygen instead of carbon dioxide. Why? Because of concentration of oxygen is more than carbon dioxide. Then this RuBisCO act as oxygenase, and here RuBisCO act as carboxylase. Then it involved to formation of one molecule of 3-PGA. Then this 3-PGA involved in C3 cycle, and also there is a formation of one molecule of 2-PG phosphoglycerol or phosphoglyceric. So it is involved in C2 cycle. Okay, phosphoglycolate will be formed here. So one molecule of 3-PGA, one molecule of 2-phosphoglycolate will be formed in here. When the concentration of oxygen is more than carbon dioxide, then RuBisCO act as a oxygenase enzyme. So in this photorespiration, who play important role means RuBisCO. So RuBisCO is the only causes or reason for occurring photo process. Okay, right. So now, so what are the conditions for here photorespiration? The condition. So when condition means when the photorespiration process will occur means. So here we will discuss in detail. So condition for photo. So generally here we will write here. Yeah. So photorespiration occur in C3 plant only. Very important. Photo occur in C3 plants only. Because why C3 plants only involved in photorespiration means, because when the temperature rises here, when temperature rises or more, or during dry condition, temperature rises or during dry condition, the plants of C3 plants, this C3 plants close their stomata. Very important. C3 plants close stomata. They close their stomata. If they close their stomata means to prevent water loss from the stomata. Water loss means here transpiration prevent. They prevent transpiration process or water loss by closing of their stomata during high temperature or during dry condition. When they water become loss means they prevent water loss or transpiration. It means that there are much water are present inside the water in C3 plants. So this cause this due to presence of water inside the plant due to prevent this water loss. So this causes two important things. One is in which due to presence of high water level in the plant body, then it cause in which carbon dioxide level inside the leaf will become decrease. So here carbon dioxide level inside the leaf become decrease. Very important, decrease. So when the much water which is present inside the leaf, then the carbon dioxide level become decrease, and very important, oxygen level will be increase. Oxygen level will increase. So carbon dioxide level decrease and oxygen level increase due to much water present inside the leaf. Why? Because leaf close their stomata, so hence there is a much accumulation of water inside the leaf. So why means all these factors. And under this high oxygen level and low carbon dioxide level, this RuBisCO act as oxygenase enzyme. They function as a oxygenase. So simply when they act as a oxygenase, then they bind to this RuBisCO enzyme binds to the oxygen. Then it binds to the oxygen instead of carbon dioxide. Once this RuBisCO bind to the oxygen with RuBP like this, RuBP plus oxygen in presence of this RuBisCO enzyme, when it act as a oxygenase, when high oxygen level and when low carbon dioxide level, then it become form into two compound, each one molecule of 3-PGA and another one molecule of 2-PG phosphoglycolate. Like that will be formed. So like that photorespiration process will occur due to all these reasons. Okay, right. Now why photorespiration does not occur in C4 plant, we will discuss here. Another slide we will go through this. Yeah. So here photo does not occur in C4 plant because the C4 plant have evolved or developed a special mechanism. So that is cranch anatomy in leaf. Cranch anatomy. So we discuss about in-depth information on cranch anatomy in C4 plant. So please watch that video. I have provided this video link for here. So because of cranch anatomy in leaf, so it doesn't occur. So likewise, and due to presence of this cranch anatomy, here what happened? So this helps in to increase the carbon dioxide concentration instead of oxygen. So which helps to increase carbon dioxide concentration. So obviously, so when the carbon dioxide concentration is more or increase, definitely what happens in RuBisCO enzyme which binds to the carbon dioxide to the RuBP in RuBP by carbon compound. So when RuBP which bind to the carbon dioxide in presence of this RuBisCO, and in this condition the RuBisCO act as a carboxylase enzyme, and then ultimately it produce two molecule of 3-PGA. So hence they not here. I'm sorry. So not here RuBP. Generally, if carbon dioxide increase, then in C4 plant generally what happened here RuBP also occur in C3 pathways. In C4 cycle, it is occur in C3 pathway like this. When carbon dioxide is much, then it enter into Kelvin cycle. So all these process of carbon dioxide fixation occur in bundle sheath cell. Why? Because the C4 cycle occur in generally two pathways. C3 pathway. C4 pathway occur in mesophyll cell and C3 pathway occur in bundle sheath cell. So this carbon dioxide fixation which occur only in bundle sheath cells, in bundle sheath cell only this RuBisCO enzyme act as a carboxylase enzyme. So that carboxylase enzyme which bind the carbon dioxide to the RuBP, then it enter into Kelvin cycle like that. Okay, so that's why this photorespiration process it does not occur in C4 plant because of the important concept here. They developed a special mechanism called cranch anatomy. So this cranch anatomy helps the C4 plant to increase the carbon dioxide concentration like that. Okay, right. Now we will go through photorespiration pathway. So here, how photorespiration process will generally occur. Photorespiration process. Photo pathway. Yeah. So this photorespiration pathway we will discuss in detailed steps. So generally here in photorespiration pathway there are three cell organelles are involved. Very important. Three cell organelles are coordinately involved. So those three cell organelles we can remember like a CPM. It is a nationalist party. CPM, Communist Party. Okay. Here C represent chloroplast cell organelle and P means Peroxisome cell organelle and M means mito. So these three cell organelles together involved in photo process. So how these three cell organelles are coordinately involved in this pathway of photorespiration we will discuss in detail. First we will discuss so what happened in chloroplast. So in chloroplast, in the chloroplast. So here in the chloroplast the RuBisCO enzyme which binds to RuBisCO enzyme which binds oxygen to RuBP. Comments like this RuBP 5 carbon compound which bind to the oxygen in presence of RuBisCO act as a oxygenase enzyme, then produce two different molecules of product. One is one molecule of 3 carbon compound called 3-PGA phosphoglyceric acid and another molecule that is also one molecule of 2-phosphoglycolate 2-PGA 2-phospho. So this phosphoglycolate has 2 carbon and 3-PGA has 3 carbon. So like that 5 carbon compound here RuBP will form. So after formation of this 2-phosphoglycolate, so this 2 carbon compound here this phosphoglycolate has 2 carbon compound and gives the cycle name as a C2 cycle. Hence we can use the term C2 cycle because in which one molecule of 2-PG will be formed and it has the 2 carbon compound. Okay, right. So once this 2-phosphoglycolate will form and this 2-phosphoglycolate here, this 2-phosphoglycolate will undergoes dephosphorylation process. Very important. It undergoes D process means removal of phosphate from PG. Then it become form into glycolate will be formed by removing of phosphate from the 2PG, 2-phosphoglycolate, and undergoes dephosphorylation process, and then it become glycolate. Okay, and the formed glycolate, very important, the formed glycolate then leave the chloroplast. This formed glycolate which leaves the chloroplast and then enter into next cell organelle, then enter into next cell organelle, that is peroxisome, then enter into peroxy. So now we'll discuss, so what happened this glycolate in peroxisome. So in peroxisome here, yeah, so in peroxisome here, second step in peroxisome, in the peroxisome, yeah, here. So in the peroxisome, this glycolate which undergoes oxidation, very important, which undergoes oxidation in presence of enzyme. Here I am writing this side enzyme, that is glycolate oxidase enzyme. In presence of enzyme called glyoxidase enzyme. In presence of glycolate oxidase enzyme, this glycolate enter into oxidation process. Then it helps in formation of glyoxylate. So along with glyoxylate, there is also formation of one toxic product as a byproduct. So that is hydrogen peroxide, H2O2. And this H2O2, hydrogen peroxide, here I am writing hydrogen peroxide, which is formed as a byproduct. And the real product is glyoxylate, but the byproduct is H2O2, that is hydrogen peroxide as a byproduct, and it is a toxic product. Very important, it is a toxic. Now later this hydrogen peroxide toxic product which broken in or destroyed, or which break into or broken or which is destroyed by in presence of an enzyme, very important enzyme here. So this hydrogen peroxide which break down in presence of enzyme called catalase, very important, catalase. So catalase enzyme is play important role to remove this toxic byproduct, that is hydrogen peroxide into, they make them into hydrogen and oxygen. So in presence of this catalase which break the or which destroy the or which break down this hydrogen peroxide into useful product like water and oxygen. Then later this glyoxylate in peroxisome only, which undergoes a process called amination process, very important, amination process. So amination means gain of ammonia group. So this glyoxylate which gain the ammonia, so NH3 addition of ammonia or gain of ammonia to this glyoxylate called amination process. Then this glyoxylate turn or form into glycine, very important, glycine amino acid get formed from this glyoxylate. When this amination process will occur, glycine will form. So when generally the formation of glycine occur in peroxisome only. So then this formed glycine then which exit the peroxisome, then later enter into next cell organelle. So that is enter into mito. Now we will discuss, so what happened this glycine in mitochondria. Yeah, so in the mitochondria, here I am writing in the mito. So in the mitochondria, these two molecule of glycine here. So two molecule of glycine. So remember each glycine has two carbon compound only. If there is a two molecule means 2 to the 4. So it means 4 carbon compound of glycines are there. And these glycine enter the mitochondria as usual. And these glycine in mitochondria, they are interact to each other. These glycine, two molecule of glycines, they are to each other, and they form one molecule of 4 carbon compound. Very important. And in mitochondria, these two molecule of glycines, they are interact to each other, and then they form the one molecule of 4 carbon compound, that is, sorry, not 4 carbon, 3 carbon compound, that is serine, 3 carbon compound. And generally glycine, two molecule of glycine has 4 carbon, but there is a formation of 3 carbon compound serine, but one more carbon release in the form of carbon dioxide. So like that 3 plus 1, 4 carbon here. And here there is a important process will occur to conversion of glycine into serine is also another amino acids. But the glycine is 2 carbon compound amino acid, serine is a 3 carbon compound amino acid. But there is a 2 molecule of glycines are there, and these 2 molecule of glycine converted into 1 molecule of serine. So hence there is a formation of 3 carbon compound called serine, and one more carbon evolved in the form of carbon dioxide. And here there is a most important process will occur here in this process, conversion of glycine to serine. So in which oxidation process will occur and also decarboxylation process will occur, decarboxylation, and here condensation process occur, condensation process also will occur in this process to convert the glycine into serine, remember. So after formation of this serine in the mitochondria, then this serine which return back to the chloroplast. How they are written means we will discuss. And so here this serine, one molecule of this serine later which return back to the peroxisomes is written back to the peroxisome in the form of hydroxypyruvate. Very important. Hydroxy in the form of hydroxypyruvate, the serine which return back to the peroxisome. So in peroxisome only this hydroxypyruvate which undergoes reduction process, very important, which undergoes reduction process, then it become glycerate, and these glycerate then enter into chloroplast, very important, then it glycerate enter back into chloro. So in chloroplast this glycerate undergoes phosphorylation process, very important. Phosphorylation process means addition of phosphate from ATP to this glycerate, then it become 3-PGA. Then again it formed back as a 3. Now this formed 3-PGA later enter into C3 cycle or Calvin cycle for production of return to continue this cycle again. So likewise in mitochondria the glycine is converted into serine. So after formation of serine, the serine returned back to the peroxisome in the form of hydroxypyruvate, and this hydroxypyruvate undergoes reduction process, then it become converted into glycerate, and this glycerate return back to the chloroplast. So in chloroplast this glycerate undergoes phosphorylation process, then it become formed into 3-PGA, and the formed 3-PGA then later enter into C3 cycle or Kelvin cycle to continue this cycle again. So likewise in the mitochondria will occur like this. So here the key points to remember in this whole steps, here I'm writing, yeah. So here important key points to remember in this photorespiration or C2 cycle, here no synthesis. So till now we discussed so many things about photorespiration. There is no formation of any kind of molecules which is useful to the plant. Like in here there is no synthesis of ATP occur, no synthesis of NADPH occur, and no synthesis of sugar also occur by this process. So no ATP, no NADPH, no means in the form of energy, and no sugar, even food also not prepared. Hence this process is known to be as a wasteful process. And very important one more, it result in the instead of producing all these things, and here this due to photorespiration they lost the carbon in the form of carbon dioxide. Hence they said to be as a wasteful process. If they lost carbon in the form of carbon dioxide, so in C3 plant the product or the yield is much lower. So yield in C3 plant, yield of the C3 plant get much lower than C4 plants. So why C3 plant produce less productivity means due to this photorespiration process. Okay, why? Because it loses carbon in the form of carbon dioxide instead of fixing it. So if they lost the carbon dioxide, definitely the productivity on this photosynthesis will be less compared to the C4 plant. So these 2 points are very important. Why? So by occurring C2 cycle in that is photorespiration in C3 plants means, so because of these important things. So no synthesis of ATP, NADPH and sugar, and due to loss of carbon in the form of carbon dioxide, the C3 plants yield is also less, okay, compared to the C4 plant. So and we will discuss all these 3 important steps in a flowchart or in the form of diagram. How exactly this will occur, we will discuss in the form of flowchart. So here I will draw 3 important diagram. So 3 cell organelles diagram like this. One more cell organelle. Yes, one more last. So likewise we have 3 cell organelles they involved. So here the first cell organelle chloro, and this one peroxisome, and this one mito. So remember these 3 cell organelles in sequential only, CPM. Okay, right. So what exactly happened in each cell organelles we will discuss in details. So in chloroplast, generally what happened here, see. So in chloroplast, 5 carbon compound REBP bind with oxygen in presence of RuBisCO enzyme, when it acts as a oxygenase, then it helps to formation of one molecule of 3-PGA, 3 carbon compound it has, and one molecule of 2 carbon compound, that is PG, phosphoglycolate. Now this formed phosphoglycolate which undergoes dephosphorylation in chloroplast only. Dephosphorylation process, then it become formed into glycolate. So here in glycolate will be formed only in chloroplast. Now later what happened? The formed glycolate, then it is entered into peroxisome. So in peroxisome, this glycolate here in peroxisome, so this glycolate, then it undergoes oxidation. So here oxidation process occur in peroxisome only, very important. So when the glycolate undergoes oxidation process, and there is a production of two important component like gly will form, and also formation of H2O2, hydrogen peroxide as a by, and this hydrogen peroxide will it is a toxic and it is break down or destroyed by the catalase enzyme. Now then coming to this glyoxylate, so this glyoxylate formed in peroxisome now. So this glyoxylate, then it undergoes amination process in peroxisome only, which undergoes amination process, gain of ammonia group, this glyoxylate. So then it result to formation of glycine. Here in peroxisome only glycine will form amino acid, 2 carbon compound, 2 molecule of 2 carbon compound glycine will form only in peroxisome by amination of glyoxylate. So once this glycine will form in peroxisome, generally glycine form in peroxisome. So after form, then this glycine enter into mitochondria. So here glycine enter into mitochondria, 2 carbon compound and 2 molecule of glycine will enter into mitochondria. So in mitochondria, so this glycine will undergo most important process like oxidation, decarboxylation, condensation. Here, so this glycine will undergo, here I am writing, so oxidation process and the process and also condensation process. Then this glycine become converted into one molecule of serine, and it has 3 carbon and one more carbon in the form of carbon dioxide. Means there are two important components will form from glycine. One is one molecule of 3 carbon compound serine and one molecule of carbon dioxide will be formed. Okay, right. So like that, here the carbon dioxide will be lost in this mito, and then this serine, what happened? This serine return back. This one molecule of 3 carbon compound serine will return back to the peroxisome. So here serine will return back to the peroxisome. Serine is also amino acids. Okay, glycine and serine both are amino acids. 3 carbon, serine has 3 carbon. See this 3 carbon serine, then which modified into in peroxisome, it converted into hydroxypyruvate. Here I am writing hydroxypyruvate, and this hydroxypyruvate later convert back by reduction process. This hydroxypyruvate get converted into glycerate. So glycerate also formed in the peroxisome only. So once the glycerate will form in peroxisome, now this glycerate will return back to the chloroplast. So in chloroplast, this glycerate which undergoes this glycerate, here I am writing this glycerate which undergoes dephosphorylation process. Not dephosphorylation, phosphorylation process will undergoes phosphorylation. Here dephosphorylation, but here in return back there is a phosphorylation process, remember. So due to phosphorylation means addition of addition of phosphate from ATP to this glycerate, then it become converted back into 3, and this 3-PGA formed 3-PGA later enter into Calvin or C3 or Calvin cycle to continue again these steps or this process. So likewise, also we have in detail steps or pathway in C2 cycle or photorespiration. So all this process in this diagram or flowchart we discuss in detail in notes just now. Okay, so this is how they coming and how they return back. We will discuss, and I explain only in this flowchart or in this in the form of diagram. That's why I wrote like this. Okay, so that's it for today class about each and every concept regarding to this C2 cycle or photorespiration, are also called glycolate pathway, are also called oxidative carboxylation process, oxidative photosynthetic carbon cycles. Okay, so that's it. And here I have provided 5 MCQs. Why? Because and this is the most important topic, so frequently they will ask number of MCQs from this topic. So here, so read all these 5 MCQs, and all these 5 MCQs which entirely covered what we have discussed just now. Okay, right. So read this. Photorespiration is initiated by the oxygenase activity of RuBisCO. So which of the following conditions strictly favor this pathway over the Calvin-Cyclin cycle? So if you read these 4 options, you will answer it and drop your answer in comment box. And coming to second MCQs here. So during the photorespiration pathway, the processing of glycolate and glycine occurs sequentially in specific cellular compartment. Choose the correct sequence of organelles involved in pathway. So just now we discussed CPM. So by that you can answer it. So remember, the involved cell organelles must and should be in sequential order. So you should remember in each cell organelles what happened. Yeah. One more MCQs, third MCQs here. So in the peroxisome, here asking specifically about peroxisome, middle one. So glycolate is oxidized to glyoxylate and producing a toxic product as a byproduct. Just now we discussed. So which enzyme, which specific enzyme which destroyed this hydrogen peroxide? Like here there are 4 options are there. So remember it. And coming to fourth MCQs from this topic only. Yeah. Review the following statement regarding the photorespiration. So here there are statement 1 and statement 2. Read this statement 1 and 2 and which of the options correctly evaluate this statement. So very easy statement here we have. And the last and most important MCQs. Yeah. So when RuBisCO binds to oxygen instead of carbon dioxide, RuBP is cleaved into which of the following product? Just now we discuss. So what he is asking here asking. So when here RuBisCO means RuBP 5 carbon compound. Yes, ribulose 1, 5 bisphosphate. RuBP means ribulose 1, 5 bisphosphate. So this RuBP is bind to the oxygen instead of carbon dioxide in presence of RuBisCO act as oxygenase enzyme. So then it form what? So what are the product which form by binding of oxygen to the RuBP in presence of RuBisCO when it act as a oxygenase? So here you have to identify which products are formed. Just now we discussed there are 2 product will form. So choose the right one and drop your answer in comment box. So that's it for today. So thank you. Have a nice day.